Binding of antigen leads to clustering of antigen receptors on lymphocytes. All cell surface receptors that have a signaling function are either transmembrane proteins themselves or form parts of protein complexes that link the exterior and interior of the cell. Many receptors undergo a change in protein conformation on binding their ligand. In some types of receptor, this conformational change opens an ion channel into the cell, and the resulting change in the concentration of important ions within the cell acts as the intracellular signal, which is then converted into an intracellular response. In other receptors, the conformational change affects the cytoplasmic portion of the receptor, enabling it to associate with and activate intracellular signaling proteins and enzymes. The antigen receptors on lymphocytes transmit a signal when they bind a ligand that causes them to cluster together on the cell surface. The requirement for receptor clustering was first shown experimentally in somewhat artificial systems by using antibodies against the extracellular portion of the receptor to mimic antigen binding. Antibodies specific for the B cell receptor or the T cell receptor activate signaling by inducing clustering of the receptor complexes. This is a very convenient system for the analysis of early events after activation, as all the cells in a sample will be stimulated at the same time, making the course of the response easier to follow. Antigen receptor clustering occurs when the receptors are cross-linked to each other. The importance of cross-linking was shown by comparing the response to stimulation with antibody FAB'2 fragments, which have 2 binding sites, with FAB fragments, which have only 1. On lymphocytes treated with FAB fragments, the antigen receptors do not cluster, and the cells make no response, whereas on lymphocytes treated with FAB'2 fragments, the receptors become dimerized and the cells respond, although they may respond only weakly. The response is strongest when the FAB'2 cross-linked dimers are further clustered using anti-immunoglobulin sera directed against the FAB'2 fragments. The extensive cross-linking of the antigen receptors that then occurs delivers a very strong signal to the cell. And that's binding of antigen leads to clustering of antigen receptors on lymphocytes. Have a great day.